of the nematode. But if the egg did not hatch, of course there is no damage to our crop. Right, the selector tested on the egg. Therefore, this formulation, they were diluted into about 4 cylinder solution, 75%, 100% the pure solution, that's stock solution. We have 75% dilution, 50% dilution, we have 25, and we have 25. That is by mixing different solution, that is 75 ml of the stock to 25 ml can all see the cereal dilution. Then the extracted eggs, that one we extracted from the gullet of the celosia. So dispensed into the 50 eggs were dispensed into the plastic container in the laboratory, and 10 ml of each of the cereal solution of the Niles, Riton, and Ractin were added to the eggs, and water was added as control. The treatment and control were regulated at average temperature of 29.5 degrees centigrade and humidity of 75%. The experiments were set up for 5, 10, and 10 days for both were pesticides. The experimental design was completely randomized with 9 treatment and 4 techniques. Criminal charge were counted for 5, 10, and 15 days. The second trial was also conducted without any notification. For the mortality that for the juvenile, the freshly egg of every gunner were incubated in the laboratory for 27 days for 5 days just to collect the juvenile. Aliquot of 10 ml of juvenile suspension contained 50 eggs of the 50 second stage juvenile. This nematode were dispensed into the plastic container, and 10 ml of each pesticide were added, and the seawater was also added as control. By adding this will reduce the effective of this pesticide to half. That's why we have 12.5, 25, 35.5, and 50%. The later design was complete randomized with 9 treatments replicated 4 times. Nematodes were observed after 5, 10, and 15 days, and number of dead nematodes were recorded for each day. For the screen-out study, soil was sterilized at 90 degrees centigrade for 5 hours. Total of 26 spots were each filled with 10 liters of steam sterilized soil. Carbofora, we want to compare this pesticide with carbofora. Still, you know that carbofora is not that good for us to control this nematode. They are very flexible. You know the application of this carbofora in our health and environment. You have to test this by pesticide. So I'm using it. Carbofora has 1.5 and 2.5 kg active ingredient per hectare were applied 1 week before planting to 4 pots each separately. 2 seeds of cucumber that market more were sown in each of the 28 pots. 1 week after germination, the seedling were thinned to 1 seedling per pot, were each inoculated with 10,000 of the hemicobacter eggs. 20 of each of the pesticide that is formulated were Nile, Liptin, and Nile were added at different concentrations at 25 and 50%. Immediately after the inoculation to 16 pots. The remaining 4 pots served as uninoculated control. Experimental design was completed and randomized design with 4 replicates. The data were collected on weekly on band length, number of leaves, and band at the end of the experiment. And at the end of the experiment, raw indexes, quantities were also taken, tomato reproduction, yield were also taken. All data were transformed using square root transformation before analysis using ANOVA, and means were separated using Tonka multiplication at 5% level of probability. Results. So we want to show the effect of this barbeza or the egg of the nematode. You can see the concentration. That's I said I want to reduce the concentration. This is at 5 days, 10 days, and 15 days. You can see that none of the eggs were hatched from the fifth day. But the water control can see as like 8 of the 50 eggs that were the garden were hatched. Likewise, the 10 days. On 10 days, too, we can see that just is not really hatched, but for water, we have 16 of them. On the 15 day, you can see that only 19 were hatched, but we can see that none. We can see that we have almost like 90 issue of egg. Whereas for water, it's only 19.13% in issue. For the juvenile mortality, see that on the water, we can see that none, we can see with on the 50, we can see that 5 of them were they are still alive. No five. For reading and writing, you can see that more of the nematodes they died in the reading and writing, irrespective of the concentration. You see them. In the seawater, only 5 content died compared to what the last the 10 days after, actually all the children are all they are not alive. See that only in water we have 72.45 and we are still alive. But for living around, irrespective of the concentration, they are no more alive. For the spin-out experiment, you can see that Carbofora, sorry, Carbofora at 2.5 kg had the highest mean fresh shoot weight compared with reactivity, which is really compared is comparative with compared with the it's not exactly different from the one with Bractin and Litor. You can see that they have almost the same threshold weight. Likewise, the number of food harvested from the plant, each plant, Carbofora weight. Likewise, the number of food harvested, they will not harvest much on this. The nematode population in the soil, in the root, and the total nematode population, they all follow the same trends. God indices and reproductive factor is really the factor of the nematode infection. You can see that God indices, if the God indices is greater than 2, reproductive factor is greater than 1, you know that nematode has got a lot of damage on that crop. But if you see on the little protein and carbofora, you can see that the God indices is less than 2. Yeah. It's less than 2, and reproductive factor is less than 1. When you compare the inoculated control, which is more, that is 4.88 and 2.39, which is greater than 2 and greater than 1. Discussion. Niot formulated biopesticide, that's Niot, Lipton, and Lactin, very effective in inhibiting egg hatch of egg recombinator and survival of second stage juvenile of the nematode at all the concentrations tested. The biopesticide reduced the nematode population in the soil and increased the growth and yield of cucumber compared favorably with carbohydrate. The effectiveness of this biopesticide might be due to active ingredients such as xylon in lepton and azathioprine in grafting. Conclusion. In view of the above findings, extension officers and farmers are encouraged to avail themselves of the opportunities of a lematicidal potential of this biopesticide that is now formulated lipton and niot vaccines in addressing nematode population on the farm. These biopesticides are less harmful to human beings, leading to no residual effects on the environment as compared to conventional pesticides. It diagnoses quickly, is effective in small quantity. However, further studies still required to test the efficacy of this pesticide on the field. The combination of the control measure, the comparative model, you remember all the control measures are stated initially, if you if you if you put it in a combination that it will prevent the population of these farmers eating enemies from reaching economic injury level. Economic injury level is the level where these nematode cause massive damage to our crop, thus increasing the yield, farmers' income, and reducing poverty in order to achieve effective and sustainable development. Recommendation. There is need to monitor these hidden enemies in the soil that they might have been present in large population with experience. During my PhD, when I used a plot, I noticed that that plot is heavily infested with open nematode. And I used the plot for my experience. After the experiment, after the foundation experiment, I left the place. That's one reason why before you use your crop, before you use your land, make sure you know the history of your land. So Dr. Fiske now went and used the, the same plot for his crop, that is tomato. He could not get anything from that crop. That is very, very necessary to know the history of your soil before you plant anything. So the population may not too there if you can plant or not, or you want to control it or manage it before you plant your crop. It's very, very necessary. And we thank God for some educated farmers now coming, they are bringing their soil to Niles, just maybe the matter are there or not before they go ahead for the planting. Is therefore an urgent need for extension officers in the State Ministry of Agriculture, Agricultural Development Project, ADPs and 9 extension staff to enlighten farmers on the presence of plant and tick nematodes in their farms and the application of crop growth and yield. There is also need to conduct farm trials in form of demonstration plots for the management of these hidden enemies using now formulated pesticides and management. And this month, I would like to acknowledge the God Almighty for sustaining me for being alive today to present this seminar. I also acknowledge the members of the National Management Committee, led by our Executive Director, Dr. Mohamed Atenda. I acknowledge all your leadership role in terms of dedication, communication, decision making, integrity, trust, empathy, and flexibility. All this was combination. I also acknowledge my mentor, Dr. E. Abdul for the knowledge imparted on me, sir. Thank you very much. My colleague, I acknowledge you. Thank you very much. Active research program staff, thank you for your cooperation. Thank you for your love. Also, the NIL staff, the academic and non-academic staff, I appreciate all of you. My husband and my children for their ending love and their cooperation. I say thank you for and God bless you.
Let's keep those hands going as the presenter takes a seat. Let's keep those hands going. Let's keep those hands going. Let's keep those hands going. Brief, concise seminar presentation by Dr. Bukola Rukayara Taiwo. Thank you so very much, ma'am. Before I hand over to the chairperson, if I can see, I would say I'm having PTSD right now. Because this course, I don't know, Gani, Gomita, Presidentors, Ayo, Bakes, Elikotileku, Kutelolema, number one compulsory, I will never forget. So I will still be this thing again. Another round of applause for the presenter. Thank you so very much. We'd like to hand over to the chairperson for comments and guiding this question and answer session. Dr. Kido.
Thank you very much, Mr. M. Thank you, Dr. Amido, for that brilliant presentation. It's very important when a scientist presents and the audience also follow and flow with her. It was really quite explicit. I'm sure you'll agree with me that we all got one thing or the other from this presentation. Very precise and very concise, very direct, to the point, and you know what she has been able to achieve at the end of her research. I'll just run through some highlights from her presentation. What caught my attention from her first two slides was saying that these hidden enemies, farmers' hidden enemies, are invisible and invincible. And you know what that means? When something is invincible, very powerful, very strong. So they're very difficult even to mitigate. And we also know that cucumber is also an economic crop. It's very important for smallholder farmers. They generate a lot of income from growing this crop. And for her to go through this, identify this nematode that reduce yield, is very key. And we can also see the damage she mentioned that is caused by this pests. There's a lot of huge losses from between 11 to 100%, even depending on the crop. And for now, the world is kicking against using a lot of chemicals because they are quite damaging to human health and to the environment. So they are trying to promote biopesticides or biological controls or even horticultural and resistance to these crops. And so we want environmentally friendly methods, interventions to mitigate against these hidden enemies. We can also see that, I'm happy to see that 9-hot Lipton and Ratkin, is being used inside 9-hot, in-house. Because when it was developed, it was targeted at tomato Ebola, which was a tuta absoluta. And so this just confirms that it has a broad spectrum. It can also be used for other pests and other crops. So this is very, very important. And it's a good success story for 9-hot. I'd also like to mention that looking at her results, it's very comparable to the commercial pesticide, that carbofuran. You can see that there wasn't much difference in terms of significance between the efficacy, of this 9-hot Lipton and Ratkin as compared to carbofuran. So that is quite encouraging. That means we can, begin to introduce this to farmers. And it was also important that he worked on, the in vitro and the in vivo methods. And she's proposing to go to the field on farm trial. And that will confirm some of these results. And in her recommendation, she also mentioned that before farmers go to the field, it's important that they know the history of their farm or that soil. And thank God for awareness now and education on these farmers. They know that it's important to do some pre-testing before going into plant crops on particular soils. And I'm happy to hear that farmers are coming now with their soil samples for analysis of nematode loads on these particular fields. So I'm very happy that this is, a very good research work. And we're happy that this is happening here in 9-hot. Once again, congratulations, Dr. Amido. So we open the floor for comments, for contributions, for questions. And I would like to give this to Edie now. Edie has the floor.
Okay. Thank you, Madam Chairperson, for permitting me to talk first. I stand on the existing protocol. I am distinguished and the colleagues. Honestly, I'm going to tell Dr. Francis, you deserve a vacation. Because without bragging me, I will tell Dr. Francis tonight. Take Dr. Abdul, I will vacation. So I'm going to watch his works this Christmas. Especially, I will advocate for it. Not the name anytime. Twice, they are going to Paris. Honestly, I enjoyed this place. I thought I was when I was in the secondary school and later first part of our university day. I was the only one that hated the market, so I don't know I have a friend in the honestly, I hated that course. Anything in markets because I can't be working on something I don't see. They are more of abstracts, but it's only today I know that they are visible but invisible. And they are minute, but their impact is magnificent. So honestly, you've done a thorough job here. I'm happy I have colleagues like you here. Uh, this is an excellent work. The manner of presentation, expert, and also you are using our products patented patented on different, and then you are coming out with another thing, which even then leads to another patent. Because this is a serious work, and it's very impactful and meaningful. And also, we need also to thank the NIHOT for even coming out with this type of product, and most particularly the 230 headed by Dr. Oleg. I think this is a very great job, and Dr. Aminu Tawa front row that we can see achieve a lot of things from this products, this magic product. I enjoyed the lecture personally, and I've learned a lot of things. And I believe also we're going to take cue from what this extra uh, explorative research, what we have inward, look at it, and then we just don't do it and abandon it. I think we need to do that. I think this be a good take for the meeting I had with HOP Syndromes on Monday. Yeah, with Dr. Yedidi. And I think I've forgotten the second person. I'm sorry, the person is seated here. I think this product also could be used in your pathology. I don't know how many by the yeah, but it's not here. But I think they can use it from what you have come out with now. Because I was told they've done a lot of field survey over several years on certain things on citrus. And if they can bring this one into it, I think it may change the narrative. So I wait to see more of this and I look forward to see. So I will not ask my question because my first question you said the chemical is cost effective. So I just quickly put it there. Then what is the alternative? And I think you provided the alternative. The only thing that remains to do here is to put it to logical conclusion. I don't know, maybe this is ongoing research. If it is, better for. If it's not, uh, it can be required into appropriate program or department or unit so that we can finish that. And honestly, you've overthrown me with most of this presentation or what I present here. So I have nothing to say that this is a very good work. And I believe
Thank you very much, sir, for that, for those beautiful comments. So we'll move on to our IMC. Ask if you have any questions, comments. Okay. IMC.
Good morning, ED. Good morning, colleagues. Good morning, staff. I will say congratulations, well done, Mrs. Aminu Taiwo. I see you in a very different mood. Very gentle the way you talk. Not that you are quiet, but then when it comes to some action, you may not be gentle. On the lighter note, I remember when we went for germplasm collection on tomato proteins. And being the coordinator there, she chairman the occasion that I shouldn't follow them. I said, no, I have to follow. Or gas sit down, you don't need to follow them. I said, why not? I have to. But at the end of the day, she enjoyed because I was able to let her know some things you have to do when you are doing jam planting. That's it. My comment or question is the area you said that it has to be, you still have to go further study on to test the efficacy of the feed. What about the work we did on tomatoes? Was it only uh, no, the tomato, apart from solving, didn't you do any tests on the nematode? It was on the feed. You didn't do anything on the feed. But you can answer it or you want to say it now. Okay. So I'm just thinking it should be part of the feed testing if we haven't done that one before.
Thank you very much. So many travel congratulations. Well, people have said it's a brilliant presentation. If there's something that is more than brilliant, I will say give it to you. Now, thank you. I had a concept of Google shape, and I know that when you say Google shape in medicine, I think it can solve almost all the problems. That means that this our now lacting and Victor, we still have not discovered the potential. So we still need to do more work with it. And that takes me to the labels, the label of those products. Sir, I wanted to distinguish between the two. Though I was showing the farmer, what to look for. Even in the label, I discovered that you actually can see the difference in the label because the labels talks about almost the same thing. The two labels for actin and Victor. So I don't know if somebody has looked closely at the label, but let the experts come together, take in all other factors, and actually describe these products very well. Because we are seeing another potential in this thing now. I'm not an expert in that area. I don't know exactly what to do. But I know there's something that will distinguish those products and also put in the potentials for farmers to actually see the potential of this. Because we are talking about money here. If you are going to push these products very well, it's able to turn around our fortune as an organization. That is the first comment I have. I don't know if it's a comment or it's a question. Time, melt has been scoring low on farm. And on-farm is an important component of our extension, our outreach to farmers. And this is a potential area. If you're able to come up with facts on this product, we can really push it to on-farm and farm and APPs across the country to just take it as one of their on-farm. They will take the technology to rival. That's be a plus for us. So lastly, I want to congratulate you and also say that what Lydia said about you going for transition is germane. But this is a very good thing. Let me also that earlier people that have presented and the presentation that's a very brilliant presentation. Let me also add that concerning these products that are coming from NACOT, Raptin and Victor, we are also giving credence to the PDC. I think this the leopard in citrus. What Raptin and this is also being tried is showing that
something is coming because some of the menace in agricultural products, that this product is able to combat them. Well, looking at the title, I think something you concluded that work is still going. But for publication, you have to streamline. For publication, you have to streamline. That's just what I want to say. Thank you.
Thank you, Dr. Aminu Taiwo, for that beautiful presentation. I checked my time. It was done for exactly 20 minutes. And I'm tempted to add my CV, Madam Sharp. Anyway, that was a brilliant one. My question is, this is just for my own teaching. You said, of course, we know that these nematodes are not visible to the eyes. So how are you able to count the eggs when they hatch? I just want to learn. That's all. And the second one, talking about demonstration plots. I think we need to, just to make evidence, I'm sorry, to back up what Dr. Radley said, I think we need to work more on that. It's what people see, they would readily agree. Thank you.
Thank you. Standing on the existing protocol. Because we are awesome. Dr. Minister, that was a brilliant presentation. Okay. Thirdly, my vote the two. The AI here is that is eucalyptus. Coin from plant eucalyptus, that's the two. And the rattan is azadirachtin. Again, then there are others which the inventor will not relate to you. That they use. At least you have to hold on to something. Before you know it, if you're not careful, people will just adapt it. But these are just two. So there are two different echoes. Is it clear? That's this pesticides. There's something else. It goes beyond controlling pesticides. I was interacting with somebody in Abuja at the point, person in charge of sales, best officer sales. And he told me his observation. He said, this does not only control this tomato we are making us. That the sediment in the process of producing that, processing the, the process of processing that. The sediments are very rich as they can be considered as nutrient crops. Very high nitrogen, that where they applied it, the maize was fantastic. So which means it goes beyond controlling pest. Yes, in like what we have adopted it by Dr. Emmanuel say, in controlling the leaf spots and the fruit spot in 9 of citrus. And it's given us some result, even though it's not that outstanding if you compare it with the conventional um, fungicide. But there's still light at the end of the line. So by the time you go to the field, give us the result, we'll not be able to start working and say, yes, this can be used. Thank you very much. Let me say, if I can ask you one question. I'm not trying to put you on this. If you take this to IIT, the source in IIT, are you likely to get the same result? Yeah?
Thank you, sir. Thank you. I want to congratulate my friend for the beautiful presentation. It was a good one. And then asking how she came about that's what made her a dermatologist. That's the egg extraction and getting the minerals and all that. Because you cannot see it and there's a way they go about it. It's a technique on its own. So, Dr. Aminu, I said I congratulate you for the beautiful presentation. But where I'm going is that you mentioned Dr. Chris going to the field and then not having the history of what has been done before, putting in his drops, and there were issues. I'm sure that was why Oga was asking that, do you have the same results in IIT? So probably, okay, so Dr. Men, okay. You see, Dr. Men, what I'm trying to bring out is there a way that we can have the history on the land that we have in 9 months? So that I don't just feel that, okay, I want to go and do a particular experiment, only to get there and I see that what is happening to my crop. Even probably if I want to introduce maybe pests or maybe nematodes or any pest or disease of interest, the results that I may be getting may not be the exact if there are maybe committees in place to tell us what we plan to wear or what a particular experiment has been done on a particular land, so that another person will not go and run into the same mistake. Then, Doc, this to all of us. When we collaborate, it works better. And I remember that I went to Dr. Oke that time before she was away. I was trying to find out about their waste in the course of generating this thing, and probably we can have something to do with it. So she said, some other group are also making use of it. Like here in IO2, we have the mushroom spent compost. The mushroom spent compost is a very good soil amendment that there's a publication that we just put out with Taylor and Francis to that effect on the use of that spent mushroom compost, as a soil amendment on celosia. And we had the result was comparable to the normal inorganic fertilizer, and it was a good one. And some of us here, I won't mention it, have come to us one-on-one that we can look into this area, we can look into that area, but we are not coming up. For her to have gone to that far, that means she found out, okay, this is vaccine, and it's from 9 months, let's see what we can bring out, and we have what all of us are rejoicing over. Please, let's encourage collaboration among ourselves. If there are areas we can collaborate, some came, madam, gather the substance for spread for us, we'll come and pack. The thing is there, and it's facing this way, they will never come back. Dr. Aminu, did you do that? Something like that.
Thank you very much. I'll start by congratulating Dr. Aminu. I wasn't there during the presentation, but what is on the board is our area. So we can easily say one or two things. Number one, let me start with Dr. Wanguma's comments on growing of the crops due to the application of new substances. The Institute of Leather Research in Kaduna, was it? Sorry, have tried to produce fertilizer from neem because it has some soil amendment properties that we know. I don't know whether the fertilizer is still available now or they've stopped. In fact, a champion this use of neem some time ago when a company came from China trying to market their product and see how neem can be used in so many things. I was invited. There's a product called neem Asa by the company in China. Is neem in form of pesticide. You can just buy and use. How the thing went and whether it was stabilized or whether it didn't, because there's a lot of policy issues that made some of these companies to go away. So I'm just trying to bring that for you to know that it has some soil enhancement properties. Then coming to Google Shade, that you said, that is not a good one. If you have a product, even if you know that it can solve so many things, targeting maybe insects, internationally they would accept it because it's broad spectrum. And when it is broad spectrum, it can kill anything. If you watch, you will see that all those furadan, carbofuran, and so on and so forth are banned. You direct, the world is advanced. You direct your product. Maybe you know that it can do something. You don't understand putting it that. And it will do this, it will do that. Target it to either insect or disease. And I know it's very effective on insect. So target it on insect. If anybody wants to use it for anything, they can use it, but not to be made, like an official thing. That's just an advice, unless nobody will accept it. Thank you.
That was why I said when I made a comment that we are looking at another thing from this work. I've said it earlier. For international standards, most especially at the critical assessment of products, most especially you don't have Google shade. People are not about don't buy Google shade. The police is arrested. Like I say, you have one tablet, you take care of hypertension, it's not a lot of police. So that's why I said, if Dr. Like or that the work is still ongoing. At the later plateau, at the conclusive part of this work, we are still looking towards another business. If they work in collaboration with Dr. Care and a team or pesticide center, if they can come out with, okay, maybe they will say writing plus or writing herbs or whatever, or writing nematodes that will take care of those nematodes below the soil. And then we can now serve as a strong target. And from what you said, this man is better research in this area. We met 2 weeks ago in Abuja when all of us are struggling for what to eat through our institute. So he was forced to make presentation with the speaker of House of Reps. So he said they are working on it. I can't tell you quiet. He finished his presentation. It was on unofficial gathering. It was even around 11:30 tonight. You're a permanent resident of one of them. I don't want to mention it. So I said, okay, so you are the one stealing my food all this one. Because we don't make official, I think we don't have right for the money. So it was shocked. And they have a lot of money trying to come up with a product for Twitter again. I said, no, we already have this thing. It's a Professor Jeffrey. So, when they get to know I finished from Zaria, they wanted to intimidate me. He said it was when I finished, he had this Professor questions. I think you're welcome. That is not what I'm saying. So at the end of the day, he has come more than 7 times in that day. Because as the speaker was so agree with us, said, look, we already have this thing, so they don't need to demand any money for this. So what we are saying regarding that, general research, yes, they've gone far, but the fertilizer they are saying is not even longer there. They've shifted from fertilizer to this even or pesticide. They want to come up with a combination for these two types. I think it's a long-term issue. Niles have done it even before I go now, as far as 2015. So the issue at this point is that if we can have more resources to this, have focus, just like the collective service, and put one energy, we're looking towards another patent again. All we need to do is just pack it well, and then put concerted effort and let people work towards it so that we can have conclusive. Yes, it will be better as a patent. We're going to start the whole process again, and then already we have the basis. For example, now, if Toyota car or Honda, they want to do Honda for 2024, they don't want to the basis again. They have the fuel mechanism, they have the shaft mechanism. All they need to do is just look one segment of it, you know, and then the light back, the rear light, and then they rename it again. So we have the fundamental now, we have the basics. All we need to do, from what Dr. Ibu has said, into vegetable and spices that, look, we need to collaborate. We just need to, that collaboration, that like we think ourselves into very constant. So that whatever we're doing, we don't have to do it and abandon it. We need to take it to conclusion. Either that is there or it's not there, or is there or whatever. Let us take it to conclusion and then we'll continue running. We have the fundamental and the basis. And that's why you see all this automobile industry, they continue progressing. Because they have the what they call, I don't know what I call it, the secret formula. So what they need just to be applying it. I think those people in Dr. Kudaki Kudewa, I want to change the code book, I'm they just have the fundamental and they change the code and they living it and Mama Polo. So we have the fundamental. So we have the fundamental now. It's four of us now to collaborate and start building on it. Would you
Thank you, sir. Thank you, Lady Chairman, or Chairwoman of each one of the United States. Chairperson, okay. Dr. Abinu, as Dr. Ejida said, this is the fastest seminar. But I'm not moved by that. To me, since you want to quickly deliver and get out of this place. But really, well, really, it's a good work. No doubt about that. It's a good work. So you like to intimidate us with all these things we don't want to get. The ED and NC is not the only person. I just hate it. I have hatred. Most of these courses in I give another of those things. I don't like them. Why? Because they will ask you to draw what you don't even see in the microscope. That's the reason why I don't like them. So, but how be we you give us a bit of understanding, and I've seen one or two things. Area of nematode when it comes to planting and banana. So I try to understand a bit of what you are saying. But my own thing is presentation and statistics. Your tables and your treatments are very significant. And the LSD is extremely low. What is your verification? How many? 4. You need to go back to the data and look at it very well. That is one. The other one is in the area of the treatments, there are two major ones, but the concentrations are okay, they are many. They are okay to the extent that even if you want to look at the concentration of each one, you can see make the story. But for some of us that are so keen in knowing in details within each of the treatments, we believe putting them together like this, even if you want to, would have been better in figures for each parameter, so we can easily see across. That's why I asked them to show this. So subsequently, good work, but at times when you see these white people, they just do the little thing, and then if you are to do it the way I'm saying, in the next 1 hour, you keep standing, not 20 minutes. And we'll have more and more understanding. Let's see, go back to your percent CV. It's not here, it's not here. It's the LSD that is here. Percent CV. When you get the percent CV, it will tell you some stories about how good the experiment is.
Coefficient of variation.
Yes, coefficient of variation. When you look at percentage CV, it will tell you how good something is. And that one will help, especially when you are moving now to the field. It's very important. I think it's okay, but throughout you did tell us which concentration is the best in each treatment. And what, for example, is it at 7.5 concentration? And when that 7.5, for example, is better than other treatment, at what level? Because with that, you can really know there's no point using high. So, and if the two of them, maybe the three in the middle, they have the same value, there's no difference between them, then there's no point going high because it will cost the farmer more. Something like that. And you agree with me now that stating all those ones, you still be standing. Thank you.
Thank you for all your comments and contributions. Thank you, Dr. S.W.S. Akinemi. When you to the fifth, I will use that one for the duration. Thank you very much, sir. You can see that in all the professional my recommendation, I said even at the lowest concentration, that they are very, very effective. And for me, that's why I use 25% and 50% in the screen house. That's why I use 25 and 50. And both are very effective. Thank you very much. Dr. Oyedele Man, for me to count that called egg, when we extracted the egg we have a procedure for the extraction of the eggs in the laboratory. We just do the extrapolation. Just take 1 ml of that extract. From the ground root, you can see the animal show to students on IT to show the egg that this is how egg nematode just like an oak shape, like a cockroach egg. That's the shape. Is that on the microscope? And when it's also ash, you can see the juvenile sweat. I think that's all the question. For Dr. BC, you mentioned that I told them that when I finished using my plot for the nematode experiment, that I went and planted tomato, and the tomato wasn't good at all. I don't know what to plant it. I think my PhD was not very good. No problem. Thank you for all the comments contribution. And then we will be able to do the necessary work. Thank you very much.
Thank you very much, the presenter. So open the floor to other colleagues. We'd like to take questions, comments, contributions from other colleagues. But please make your questions very, very precise. Be direct. No ambiguity. So please, we don't have much time. Questions, comments, contributions, questions. Don't come with this questions. So don't be thinking like we can do it and we say you ask 4 questions. What you are doing? Please.
So we have one on this side.